previous section, we already discussed the cell-mediated immunity, which is one of the type of the adaptive immunity. And in this section, we will discuss the humoral arm of the adaptive immunity, in which we are going to discuss the mode of the activation of the B cells, effector function of B cells, what is antibody, and the structure of the antibody and its types, and the primary and the secondary response of antibodies, and the function of the antibodies. So the humoral immunity is provided by the B lymphocytes, and this immunity can recognize proteins, polysaccharide, phospholipids, and nucleic acid antigens, and it provides immunity against the extracellular bacteria, viruses, and toxins, and it causes type 1, type 2, and type 3 hypersensitivity as compared to the cell-mediated immunity, which is provided by the T lymphocytes, and can recognize only protein antigens, recognize antigens presented by the antigen presenting cells with class 1 or class 2 MHC molecule, as the cell-mediated immunity is discussed already in the previous section. And this cell-mediated immunity causes type 4 hypersensitivity and causes the acute graft rejection. So the adaptive immunity is the type of the immunity that the body develops during lifetime, and it develops after exposure to antigen. So the adaptive immunity develops slowly as compared to the innate immunity, but it is specific and improves upon the repeated exposure and has memory cells. That's why it is providing long-term protection. And again, we are going to revise. The adaptive immunity has two arms, the cell-mediated and the humoral immunity. So in this lecture, I am going to teach the humoral immunity. It is an antibody-mediated immunity, and it acts against the exotoxin-mediated diseases such as the tetanus, diphtheria, pneumococci, meningococci, Haemophilus influenza, and certain viral infections. So, as we said that the humoral immunity, in humoral immunity, there is a basically involvement of the B lymphocyte. So what are the main important functions of the B lymphocyte is that it going to differentiate into the plasma cells and produce antibodies. And as we said earlier, that the B cells is the most important, one of the most important antigen presenting cells. So the B cells can present antigen to the helper T cells. For the other two antigen presenting cells, the dendritic cells and the macrophages. So now comes the clonal selection. What is clonal selection? It is defined as that the antigens select a lymphocyte that will undergo clonal expansion and produce more lymphocyte with the same type of antigen receptor. So in clonal selection, only one type of antibody and one type of B lymphocyte responds to the antigenic determinant. And that is person has a large pool of B lymphocytes, and each B cell bears a surface receptor, either IgM or IgD, that can react with one antigen. Now comes to the mode of activation of T cell. So after the clonal selections means that after the antigen binds, lymphocyte is stimulated to proliferate and produce more lymphocyte with the same type of antigen receptor, that is form a clone of cells. Then these B cells becomes plasma cells and secrete the antibodies specific for that antigen. So same process of clonal selection also occur with the T cells. So this is a T cell independent B cell activation, in which there is no involvement of the T cell. The another mode of B cell activation is T cell dependent activation. What happens in T cell dependent activation of B cell? That is, a B cell is triggered when it encounters its matching antigen. The B cell then engulfs this antigen and then it displays the antigen fragment bound to its unique MHC molecule. This combination of antigen and MHC attract the specific T cells. And the cytokines are secreted by the T cell, help the B cell to multiply and mature into antibody producing plasma cells. And the release into the blood antibodies lock onto the matching antigen, and the antigen antibody complexes are then cleared by the complement cascade or by the liver and the spleen. So this is a T cell dependent activation of B cell. Now, what is the effector function of the B cell? As we already discussed that after activation, B cells convert into the plasma cells which secrete antibodies and fight against the infection. But some B cells become converted into the memory cells which remain inactive for long periods but are capable of being activated rapidly upon re-exposure to same antigen. Because of these memory cells, antibodies appear rapidly in secondary response. That is, they provide long-term immunity to same infection. So, this is the effector function of the B cell. Now, what is antibodies? Antibodies are the gamma globulin protein that react with the antigen that stimulate their production. So antibodies are also called the immunoglobulins. So there are certain important functions of the antibody. The most important function of the antibodies are to neutralize toxins and the viruses, to opsonize the microbes so that they are more easily phagocytosed, to activate the complement and to prevent the attachment of microbes to the mucosal surfaces. And antibodies also have an enzymatic catalytic capability. So these are all the functions of the antibodies. Now come to the structure of the immunoglobulin or the structure of the antibody. So this is the structure of the immunoglobulin G, and it is Y-shaped. Molecule consists of 2 light chains and 2 heavy chains. These are the 2 heavy chains and these are 2 light chains. Each light chain consists of a variable region and a constant region. Most heavy chains consist of 1 variable and 3 constant domains. The term heavy and light chains are according to their molecular weight. These 4 chains are joined by a disulfide bond. And the variable regions of both the light and the heavy chains responsible for the antigen binding and the constant region, so this is the constant region, and the constant region of heavy chain is responsible for complement activation because it contains the complement binding site, and it is also responsible for binding to receptors on neutrophils and macrophages. So if an antibody molecule treated with a proteolytic enzyme, its peptide bond which is present in the hinge portion will break and producing 2 identical Fab fragments which is having antigen binding site and 1 FC fragment which is involved in placental transfer, complement fixation and attachment sites for the various cells. Now the immunoglobulin classes and subclasses. So, in terms of the differences in the amino acid sequence of the constant region of a heavy chain, antibody molecules are divided into 5 classes. It is immunoglobulin G, immunoglobulin A, M, D, and E. Heavy chains are distinct for each of the 5 immunoglobulins, and the light chains belongs to one of the 2 types, it's either kappa or lambda. Now, the IgG is the main antibody in secondary response. It opsonizes bacteria, making them easier to phagocytose, and fixes the complement, which enhances bacterial killing and neutralizes bacterial toxins and viruses. It can cross the placenta. It is divalent, means that it has 2 identical antigen binding sites. Now, the IgA is the main immunoglobulin in secretions like colostrum, saliva, tears, respiratory, and intestinal secretions. Basically, the IgA prevents the attachment of the bacteria and the virus to the mucous membrane. Now, the IgM, it is produced in the primary response to an antigen, and it fixes the complement. It is present as a monomer on the surface of all B cells, where it functions as an antigen binding receptor. It is a pentamer and has 10 antigen binding sites. Now, the IgE mediates the hypersensitivity by causing release of the mediators from mast cells and the basophils upon exposure to antigen. And it is the main host defense against the helminthic infection. The IgD found on the surface of the B cells, where it acts as a receptor for antigen. Now comes to the primary and secondary response. Antibody responds to initial antigenic stimuli, and it is called the primary response. The primary response differs both quantitatively and qualitatively. The primary response is slow, sluggish, and short-lived, while it has a long lag phase and low antibody titer, and IgM is a predominant in primary response. Subsequent to the primary exposure or subsequent to the primary response is called the secondary response. It is prompt, powerful, and prolonged as compared to the primary response. It has short or negligible lag phase. It has much higher level of antibodies and for the longer duration. And immunoglobulin G is a predominant in secondary response. So this is all about the humoral arm of the adaptive immunity. Thank you so much, dear students.